everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to discuss about the immunoassay techniques. How different immunoassay techniques are being performed in the laboratory, what are antigens and antibodies we have discussed in the previous lectures, and now we are going to discuss how these techniques are competitive and non-competitive, how these techniques are homogeneous and heterogeneous in nature. So, whenever an antigen and antibody reactions take place, we have different types of techniques going on. Antigen always combines with a specific antibody, and to make this reaction, to measure the amount of antigen or antibody present, we make our certain antibodies labeled. So, during a competitive reaction of an antigen and antibody, we have one step competitive format. In this, the labeled antigen, reagent and the unlabeled specimen compete for a limited amount of antibody. The amount of antibody is few and the amount of antigen we have large. So, there is a limit, and in half of the antibodies the labeled antigen will go and will give a specific reaction, and in another half the unlabeled will go and give a specific reaction. This is known as one step competitive format of an immunoassay technique. Then we have two step competitive format. In this, the antibody concentration of the reaction is present in excess in comparison to the concentration of antigen. That means the concentration of antibody is a lot. Antibody reagent is incubated with specimen containing antigens, and in the second step labeled antigen is added. In the competitive format, less bound labeled antigen indicates more antigen present in the test sample, and this two-step competitive assay formats provide several fold improved assay sensitivity compared to one-step assay format. So, we can see here in this case the antibody is in excess, but the antigen is very limited. The certain antibodies will be free and the antigen will give certain reaction with the previous antibodies. So, in the step 2 of the competitive format, we add more labeled antigens. So, how much more labeled antigen will go and react with the antibodies will give us a measurable reaction. Now, the next one is we have the non-competitive or the sandwich method. In this format, generally it provides the highest level of assay sensitivity and specificity and are applied to the measurement of critical analytes such as cardiac and hepatitis markers. This format is referred to as the sandwich assay because analyte is bound between two highly specific antibody reagents. So, the non-competitive methods, they are also known as the sandwich method, where the analyte is bounded between two antibodies, one from this side and another one from this side. So, whenever the antigens will be in between and we have the labeled antibodies, this is a type of non-competitive type of format, and the two type of antibodies and two types of antigens are in between and they make a kind of a sandwich reactions. It is known as non-competitive type of reaction. This assay can also utilize either one step or two step methods as with the competitive assay. The two step assay format employs wash steps in which the sandwich binding complex is isolated and washed to remove excess unbound labeled reagents and any other interfering substances. So, specifically we do one step where washing is done and whatever material which has not been bounded to the antibody, that reagent comes out. And in the second step, usually it offers the highest specificity and sensitivity of all the assay formats which we have discussed before. So, in such cases, of the non-competitive assay of the second type, whenever an extra step of washing is being added, the excess unbound labeled reagent or any other substance which is interfering in the non-competitive assay reaction will be removed. So, the non-competitive assay, it is very specific in nature and very sensitive compared to the competitive techniques which are being discussed before. So, in the non-competitive assay, the measurement of the labeled sample or antigen is directly proportional to the amount of antigen present in the body, and it can be represented by a dose response curve. The x-axis shows the concentration of antigen and the y-axis shows the response. So, we can see a direct relationship between the signal and the concentration, that how much antigen and antibody is present. It is a dose response relationship, that how much dose will be added, that much response will be there. So, if more antigen and antibody will be added, then more reactions will be there, and this graph shows a direct relationship between the signal and the concentration. The higher the signal, the higher the concentration. So, after discussing the competitive and non-competitive assays, we are discussing the homogeneous and heterogeneous immunoassay methods. Those methods that require separation of bound antibody antigen complex are referred to as heterogeneous immunoassay, and that do not require separations are referred to as homogeneous immunoassay. Homogeneous methods have been generally applied to the measurement of small analytes such as abused and therapeutic drugs. Since homogeneous methods do not require the separation of the bound antibody antigen from the free antigen, they are generally much easier and faster to perform. So, in the cases of homogeneous reactions, normally they are done where the quantity of antigen is very less in cases of drugs. But on the other hand, if we talk about the heterogeneous immunoassay reactions, they have to be, the antigen antibody have to be removed away from each other, but in the homogeneous reactions the antigen antibody complex remains same. In these immunoassay, as they do not require the separation of the unbound complexes, they are quite easy to perform, whereas the heterogeneous reactions, they require the separation of the unbound complexes. So, in such cases, the heterogeneous reactions, they are little slower to perform, whereas the homogeneous reactions are very fast. As we can see in the image, that the homogeneous methods show where the antigen antibody complex is being formed, where we have antigen antibody and a specific specimen antigen, and a complex is formed, and the complex we do not have to remove the antigens or antibody away from each other, but in the heterogeneous method the ABAG complex has to be broken. That is a heterogeneous immunoassay technique. Now, these immunoassays are tests, they uses antibody and antigen complexes, which are also called immunocomplexes, to measure the presence of a specific analyte in a sample. So, as we have discussed that immunoassays are a quantitative techniques and they are very specific and have very high affinity. So, these immunoassay methods, they can specifically measure the amount of a specific antigen which is present in a reaction or in a sample. Whereas antibodies, they are just proteins which are normally produced by the immune system in response to a foreign substance, but antigens are those molecules which binds with the antibody and they make a complex if they both are specific in nature. So, antibodies are comprised of two major regions, the Fab region and the Fc region, which we have discussed, the heavy chain and the light chains. And the antibody preparations are either polyclonal antisera, that it has many arms, which recognizes multiple sites on antigens, or monoclonal antibody, which requires or which recognize only single sites on antigens. So, antibodies, depending upon what kind of reaction we have to go for, we can prepare monoclonal antibody and we can also prepare polyclonal antibodies. In immunoassays, the antibody or antigen is labeled in order to have a measurable signal which corresponds to the concentration of the analyte, which we have discussed about how these reactions can be competitive or non-competitive. In competitive immunoassay, amount of antigen is indirectly proportional to the amount of signal, whereas in non-competitive, we have seen in the graph, the amount of antigen is directly proportional to the amount of the antibody which is present, or the reaction which is based on a dose response relationship. Now, let us talk about the complement fixation test. This is also an immunological medical test looking for evidence of infection. It tests for the presence of either specific antibody or specific antigen in a patient's serum. It uses sheep RBCs, anti-sheep RBCs antibody and complement plus specific antigen if looking for antibody in serum, or specific antibody if looking for antigen in serum. So, if we are looking for antibody, then we require specific antigen, but if we are looking for specific antigen, then we require specific antibodies in the cases of a complement fixation test. If the antibody or antigen is present in the patient's serum, then the complement is completely utilized, so that the sheep RBCs are not lysed. But if the antibody is not present, then the complement is not used up. So, it binds anti-sheep RBCs antibody and the sheep RBCs are lysed. One of the major example of complement fixation test is the Wassermann test. Now, amongst these, there are certain techniques which are the example of various immunochemical techniques, where we have the competitive format, where we have the non-competitive format, where we have the heterozygous reaction, where we have the homogeneous reaction. So, one major technique which is an example of immunoassay technique is ELISA, enzyme linked immunoassay test. So, enzyme linked immunosorbent assay test, or also known as short form ELISA. It is a biochemical technique which is used mainly in immunology to detect the presence of an antibody or an antigen in a sample. The ELISA has been used as a diagnostic tool in medicine and plant pathology, as well as a quality control check in various industries. In simple terms, in ELISA, an unknown amount of antigen is affixed to a surface and then a specific antibody is washed over the surface so that it can bind the antigen. So, ELISA technique is used to know the amount of antigen which is present on that surface or in the analyte or in the sample. This antibody which we are washing over the antigen present is linked to an enzyme, and in the final step a substance is added that the enzyme can convert to some detectable signal. So, whatever enzyme we are adding, we have to convert it to some detectable signal which can further be analyzed. In the cases of fluorescence ELISA, when light is shown upon the sample, if there is any antigen antibody complex which is being formed, will give fluorescence, so that the amount of antigen in the sample can be easily measured. ELISA is a quantitative technique where we can measure the amount of antigen present in the particular analyte. So, we have different types of ELISA. The way we make changes in those ELISA tests, they are named on that basis. The first one is indirect ELISA. They have the similar steps, general steps. We apply a sample of known antigen or known concentration to a surface. Often we keep the sample in a microtiter plate and the antigen is fixed to the surface to render it immobile. Simple adsorption of the protein to the plastic surface is usually sufficient. So, in this case we take the microtiter plate. In the microtiter plate we are adding non-antigen of known concentration. That means we know that we have added this many antigen and this many concentration is present in that particular hole of the microtiter plate. These samples of known antigen concentration, they will constitute a standard curve used to calculate antigen concentrations of unknown samples also. So, in the second steps, the plate wells or other surfaces are coated with serum samples of unknown antigen concentration and diluted into the same buffer used for the antigen standards. Since antigen immobilization in this step is due to non-specific adsorption, it is important for the total protein concentration to be similar to that of the antigen standards. Now, a concentrated solution of non-interacting protein such as bovine serum albumin, that is BSA, or casein is added to all the plate wells. This step is known as blocking because the serum proteins block non-specific adsorption of other proteins to the plate. So, this is in indirect ELISA where first the antigen is being added and then antibodies are being added. After this step, the plate is being washed and a detection antibody specific to the antigen is applied to all the plate wells, but these antibodies will only bind to immobilized antigen which is present on the wall surface and they would not bind to any other serum proteins or the blocking proteins. This plate which we have prepared, it is again washed to remove any unbound detection of antibody. After this wash, only the antibody antigen complex remain attached to the wall. So, antibody which is very specific to the antigen, that antigen which is present on the outside of the titer wall, it gets attached to that particular antigen antibody complex. So, whatever the secondary antibodies which will bind to the remaining detection antibodies are added to wells now, and these secondary antibodies are conjugated to substrate specific enzyme, and this step, if it is not required, it can be skipped and the detection of antibody is conjugated to an enzyme and this step can be skipped in such certain cases. After that, we wash the plate and whatever excess unbound enzyme antibody conjugates are removed. We apply a substrate which is converted by the enzyme and which gives a chloromagnetic or fluorogenic or electrochemical signal, and it can be viewed and the quantity can also be studied using a spectrophotometer or a spectrofluorometer. So, whenever we are conducting ELISA, for the detector we have any technique which is chromogenic or which is fluorogenic or which will provide electrochemical signal. So, these signals or these chromogenic or fluorogenic things will help us in identifying the substrate as well as the quantity of substrate or the sample which is present in our analytes. That was indirect ELISA. Now, we discuss about the sandwich ELISA. The plate is coated with the capture antibody. We add the sample to the plate and any antigen which is present and binds to the capture antibody. Detecting antibody is added and binds to the antigen. Enzyme linked secondary antibody is added and they also bind to detecting antibody. Substrate is added and is converted by enzyme in detectable form. So, a less common variant of this technique, we call it as sandwich ELISA and it is used to detect the sample antigens. For this, we follow various steps. we prepare a surface to which a known quantity of captured antibody is bound. We do not add the antibody, we have to capture it on the surface. We block any non-specific antibody sites on the surface. We apply the antigen containing complex or sample to the plate and we wash the plate so that unbound antigens which are present, they are removed, as we have done in the previous cases. So, after that we apply primary antibodies which will bound specifically to the antigen, and we then add the enzyme which is linked to the secondary antibodies which are specific to the primary antibodies, and then we wash the plates so that the unbound antibody enzyme conjugates, they are being removed from there, and only the reaction is given by those antibodies which are bound to the plate. Then a chemical is applied which is converted by the enzyme into a color or a fluorescent or electrochemical signal. As we say, the reaction could be chromogenic, fluorogenic or electrochemical in nature, and then we measure using the detector which could be a spectrophotometer, fluorimeter, colorimeter, etc. We measure the absorbance or fluorescence or electrochemical signal of the plate to determine the presence and the quantity of antigens which are present in that. Next we have a competitive ELISA. It is based on the way we have studied about the competitive and non-competitive techniques. In this specific, the reaction takes place through competitive binding. The steps for this ELISA are somewhat different than the first two example. In this cases, the unlabeled antibody is incubated in the presence of its antigen. These bound antibody antigen complexes are added to an antigen which is coated very well, and the plate is washed so that unbound antibodies are removed. The more antigen in the sample, the less antibody will be able to bind to the antigen in the well. Hence, we call this technique as competitive ELISA. In the next step, the secondary antibodies specific to the primary antibodies are added, and this second antibody is coupled to the enzyme. Then a substrate is added and remaining enzymes elicit a chromogenic or fluorescent signal. For competitive ELISA, the higher the original antigen concentration, the weaker the eventual signal. So, we see in the competitive binding, we add different types of antigens and antibodies, and if the antibodies are attached to the wall of the microreactor plate where the reaction is being conducted, then we wash it for the excess antibodies which are not present. Now, the applications of ELISA. Because the ELISA can be performed to evaluate either the presence of antigens or the presence of antibody in a sample, it is a useful tool for determining serum antibody concentrations in various tests, or also for detecting the presence of any antigens which are being present there. Coming to the application, ELISA has found applications in the food industry. We can use it for the detection of potential food allergens in the milk, peanuts, walnuts, almonds and egg. The ELISA test or the enzyme immunoassay was the first screening test commonly employed for HIV. ELISA technique is highly sensitive. In an ELISA test, a person's serum is diluted 400 times and applied to a plate to which HIV antigens have been attached. If antibodies to HIV are present in the serum, they may bind to these HIV agents. The plate is then washed to remove all other components of the serum. A specifically prepared secondary antibody and antibody that it binds to human antibodies is then applied to the plate, followed by another wash. This secondary antibody is chemically linked in advance to an enzyme. This secondary type of antibody which binds to the human antibody and again which is applied to plate is removed by the washes. So, depending upon what kind of result we are expecting from the ELISA technique? We can use different types of ELISA. We can use non-competitive ELISA, we can use competitive ELISA, we can use sandwich ELISA, and whenever we are conducting it, different ELISAs, we have to be taken care of that antigen antibody reactions which we are conducting. The antigen and antibodies should be bounded to the outer wall of the plates, and whenever we are washing, only the excess should be removed out. To remove the excess, the plates are being washed multiple times and the reactions are being taken place with the antigen or antibodies which are present on the walls. The enzymes which are being used in the ELISA techniques, these enzymes, they are labeled so that the technique could be measurable in nature. So, the plate will contain enzyme in proportion to the amount of secondary antibody bound to the plate. A substrate for the enzyme is applied and catalysis by the enzyme leads to a change in color or fluorescence. So, if we are conducting a color change or a fluorescence change, we have to look whether the reaction is chromogenic in nature or fluorogenic in nature. ELISA results are reported as a number. The most controversial aspect of this test is determining the cutoff point between a positive and a negative results. So, whenever we are conducting an ELISA technique, we look for the measurable reactions, we look for the change in color, we look for the change in fluorescence. So, the reaction is called the chromogenic reaction or the fluorogenic reactions, and depending upon, we have to determine that cutoff where we have to see whether the reaction is a positive or negative result. So, to conclude about the immunoassay techniques, there are various techniques which we have discussed in the various series of these lectures, starting from the ring test, double diffusion test, single diffusion test, immunoelectrophoresis, various competitive, non-competitive methods, ELISA, radioimmunoassays, etc. that can be applied to various fields in medical as well as forensic science, and they give specific results which can help in solving a case positively. Thank you.